Welcome everyone. We are very excited about the last seminar of this semester. The speaker today is Heather Harrington, and she's going to tell us about multi-parameter persistent homology and applications.
Great. Thanks for the invitation. And yes, so I'm going to talk about kind of two different projects today. And there's something kind of in it for everyone. So there's a bit on the multi-parameter, but also, as I said, on the applications. And this is joint work that we have on the archive with Nina Otter, Hal Schenk, and Ulrike Tillman, and then some work that's relatively new with Bernadette Stolz and Helen Byrne. Okay, so just to review about single parameter persistent homology. So if we have some data, we might want to ask, well, what is the topology of the set of these points? And that's not a very interesting question because the points themselves don't have interesting topology, but as soon as you thicken and then take the union, we can study the topology of this. So how do we choose the epsilon? And so if your epsilon's too small, then you won't see any interesting homology. And then if epsilon is too large, then again, you won't see this this whole. So the solution is really to consider all possible values for epsilon. So then you obtain this nested sequence of spaces, and then you can study these topological features, and in particular how they're changing across the filtration. So this is a review. So if you're given some simplicial complex, then you can look at some coefficients that are in a field, and so you have these simplicial complex and with bases of the I simplices, and then you can form this chain complex, and we call this the nth simplicial homology of your simplicial complex K. And then we can look at the homology groups, and then the dimension of them give you the Betti number. So just a very simple example, if we're looking at this triangle, then we can see in a field with two elements that, for example, if you're looking at the boundary maps, these differentials, you have D1 of A is just this vertex plus this vertex. So then we can look at the different dimensions. So Betti 0 is the connected components, and that would be 1, and Betti 1 is 1. Okay, so that's just a review. So then when we're, so then with persistent homology, as I said, we obtain this nested sequence, and we would call this a filtered simplicial complex. And when we apply P's simplicial homology, we then have this, then we have these homology groups, but then the linear maps such that there's composition, and this is called the P'th persistent homology of your simplicial complex. This is, I think, all review. All right, kind of quick. Okay. So then homology modules of such a filtered space, they come with some additional structure. So we have these K vector spaces, and we have these linear maps modules, and this additional structure that we have are these are finally generated graded modules over a polynomial ring with coefficients that are in some field. So the grading will give us some information about the position in the filtration, and then the action of X gives the shift in the filtration by one position. Then there's the correspondence theorem. So if you're given a persistence module, you can associate to it a graded module over a polynomial ring by taking the direct sum of all these vector spaces and then the action on your on your module, it's given by the linear map. And similarly, given a graded module, then this direct sum, we can assign to it a persistence module where these collection of vector spaces are given by the summands in the decomposition. And then, so then your linear map F from Mi to Mj is given by the action. So these assignments are inverse to each other, but there's even more. So you have this isomorphism of categories. Okay, but so then I guess this is all still review. So then you have the structure theorem for finitely generated graded modules over a polynomial ring in one variable. So it's a principal ideal domain. And then you would get that you have this very nice decomposition into the free parts and the torsion parts. So these correspond to, in these intervals, these are the features that just exist for a finite interval, and then these are the infinite intervals. And this whole collection is called a barcode. So that's the complete invariant for persistence modules. And so I don't know, can you see if I put something up? I kind of have little examples as I go, but I don't know if you can see it because can you see that? No.
I can see that.
Okay, if you can't, then I can just read it out as well. Maybe it's backwards.
I don't know what you're looking at.
Oh, sorry. Who is it?
I can see your slides, but when you put up something else, I have no idea.
Okay. Well, I just wanted to. Okay. So that's not going to work. That's okay. So I was just kind of going to give examples as I go for what things look like. So I just kind of okay. So if you have okay, so I was just giving an example of a barcode.
Share your screen instead of just the application, then whatever's on your screen will show up. But at the moment, you might.
No, I just I hand wrote out some things, but it's fine. So I was basically just showing a barcode where you have it's fine. Actually, it's fine. Don't worry. Never mind. So I'll just continue. So then I was giving a concrete example. But so then what we have is this nice pipeline, persistent homology pipeline, right, where you start with a filtered simplicial complex. You start with your data and you build this filtered simplicial complex and you compute homology, and then you have your barcodes. And so we wrote a paper that talks about the computation of persistent homology, and it's made to be accessible for non-topologists. But the nice thing, I think, even for anybody who's doing computation, is we have Nina wrote code so that any input or output of any data set or complex or whatever, you can actually go between. So you can you can use it for different softwares. Okay, so that's the kind of the setup with single parameter persistent homology. So now I want to talk about some applications that I'm really excited about. So you can apply persistent homology to different types of data, so finite metric space, as I just motivated. Then you can also look at some weighted networks or images, grayscale images, for example. And so here you can use persistent homology to study grayscale images where, for example, if each of these are some pixel, you might have a value on that pixel. And then you would create a filtered simplicial complex based on this, on the value here, and then eventually fill in, kind of adding to the complex. And so this is looking at grayscale images. You can see that there's one connected component all the way through, but then also there's a hole here in the middle that eventually dies. And so the reason that one might want to study images is if we have some data of tumor blood vessel networks, which is all image data. So I'm going to talk about, this is a project that I've been interested in for a very long time, but now it's kind of we have a lot of energy. So the focus is that near a tumor, so for many years you can have an avascular tumor, but as soon as a tumor has blood vessels that grow to it, then it can vascularize and it can grow. It gets nutrients through the vasculature. So angiogenesis, this growth of new blood vessels from pre-existing vasculature, is a hallmark of cancer and also for spreading into other tissues. And in particular, we want to understand how this works. So I'm not going too much into the biology, but one of the things that we'd like to do is test what are the key mechanisms, and you can do that with hypotheses structured, encoded into mathematical models. And in particular, what we find is near a tumor is the development of loops appearing. It's called anastomosis, when blood vessels grow. And so an important question that we're working with people in radiation oncology that I'll mention in a couple of slides is where and when treatment should be administered near a tumor. So it turns out that this is not a new problem at all. Since the 80s, and actually there's work even earlier than this looking at understanding and predicting behavior in tumor-induced angiogenesis. And so as Helen Burns says, there are bucket loads of models. So more than 10 years ago, I worked on a paper where we looked at different geometries for placement of inhibitors with tumor-induced angiogenesis. And I'd like to point out in particular, there's a really, really nice review here. But even within that review, they suggest 12 other papers that are review papers of math models. And there are really a lot of different types of mathematical models. So I mean, I hope I'm setting this up that it's a really exciting biological problem where clearly you're already maybe getting a feel that topology can be useful. On top of just what I showed you, which is kind of the cellular level, there's really different levels within studying this problem. So from as you can see, different layers where you're looking subcellular all the way to tissue. But there's a lot of challenges. So understanding these complex geometries, their 3D simulations, often these parameters, it's not just PDEs, they're hybrid models, it's on lattice and off lattice. So you have these cellular automata. And then we want to compare these mathematical models, these hypotheses of how things are working with data. And so we'd like to first, the experimentalists, people spend years and entire PhDs extracting key parameters for the models. And then you also want to compare between in vivo and in vitro, so whether it's in an organism, and how do we understand the difference between these types of data, and then also noise. So, for example, I think this is really a nice, very recent paper here where you can see they're trying to clean the images, and so what appear to be holes here, they go through a whole image processing, and how do you distinguish between artifacts and real features? And then finally, we have experimental data that can be an initial condition. And so these are just two different examples of starting with some data, and that's an initial condition to put into a model. So here it's actually looking at vasculature and renormalization, and in this, it's they start with the vasculature from real experiments, and then they grow a tumor spheroid. So then when I was interested in this problem, the data looked kind of like this before. So this is a healthy tissue, and then this is a cancerous tissue. You can see there's they're very bendy, this high tortuosity, and so you can really see the difference. And so practitioners can look and see, but they'd like to quantify it, and many statistical approaches fall short. So now we really have state-of-the-art, gorgeous data by Oxford's Radiation Oncology group up the hill, and they're looking at, we have two different types of data sets. We have micro-CT, which are static. It's a static data set, so you can see rather quite deep into the tissue, and this is where they've put a tumor, a pellet of and it's mouse colorectal cancer, and they look at different categories, whether it's a control, so it's the cancer's treated or not, and then this intravital 3D, and this is a time series data where you're looking over a time course data where you're looking over dynamically how the blood vessels are changing over 12 hours, and then they have various different things that they're testing with it. So just to give you an example of the micro CT, they have this image, and then people spend their entire PhDs segmenting these images. And so just, I'll give you, it's going to work. Oh, it's a pretty video. For some reason, I guess it's not. I don't know why it didn't link, but it's a really nice video where you can see, and the changes in the color relate to different, the vessel thickness, the diameter. So you can see there's some vessels that are larger than others. So here with this multiphoton intravital 3D imaging, they can't go very deep, but they have really high resolution. So they put this dye, this fluorescent protein that stains, so then they can see, as it says, a small penetration depth. But then we can see what's happening over time, and they basically hook a mouse up to a lot of different cameras. So we have images of this. So you can see different behavior, and experimentalists are interested in different things, whether it's the blood vessels sprouting, so bifurcating, or perfusion of a vessel. So there's various different there's various different characteristics. And then we also have a really nice data set.
Heather, a question there, if I could interrupt.
Sure.
So what's the time scale for a vessel to sprout? Do you need to keep this this device on for a couple days?
So for ethical reasons, it took a long time for them to even get approved 12 hours. And you can see the vasculature changes a lot in 12 hours. Whereas in the micro CT, what they only get per mouse one image. They sacrifice a mouse for each of these images. And then there you can do it we have them at different days.
Thanks.
But for this, it's only 12 hours. Once they insert it, then you have 12 hours of imaging, and then the dye fades.
Wow, thanks.
Mm-hmm. But it's really, it's incredible because a lot of these models also are, they're changing with time. We're looking at the growth of these vessels. So, okay, and then the other data set that's really nice that we have is a Roche data set, and I believe that everything I'm talking about is published in this paper. But here, it's ultra microscopy, so they have different sections, and then they put them all together. And so these are slides from Bernadette, so I don't know much of the details for this in particular, but they get all these slices from this, and then they can somehow, can render that. So as it says here, the image tumors up to 5 micrometers and millimeters in diameter, and they have a spatial resolution of 5 microns. So it's really nice. It's very high tech with these all the imaging. Yeah. So then, the big question is, even with all of this, is how to quantify these different characteristics, and there's lots of questions. And this is from Jakob, who's a PhD student in math bio, I believe. So they have these images, and then they skeletonize it, and they are looking at different statistics. So the length of the blood vessels, of the vasculature, the diameter, and even with these statistics, it seems that we need more. So there's lots of different biological questions to ask. The one in particular that I think is really nice is, can we quantify how much blood vessels have renormalized? So when there's a tumor and they put some type of therapy, often the blood vessels go from looking kind of diseased to becoming renormalized, so they look healthy again. And so that's a question that they're particularly interested in, is can we quantify that? Okay, and so then this is some work that from Villanova et al. So and Gomez is one of the other authors, and they have looked at different types of models, and from these models, then even though each one is different, is there some common characteristics, and how when you change different parameters, how sensitive the results are to that. So yeah, so I mean, when we did this, it was 10 years ago, but we used all the supercomputers we had at the time and very complicated geometries that would take a very long time, weeks to run. So from all of this data that's generated from the simulations, can we use that to say how good the model is, whatever that means, because there's no correct model, right? All models are wrong, but some are useful. So, right, and then we would like to be able to get some measures of how it compares, for example, with the cell density or the vessel length, the diameter, as well as other measures. Can we see, for example, using some type of different descriptors can give us, like structural information in particular, that can give us some information about the oxygen distribution, for example. So then one of the large challenges that Bernadette has faced using trying to do this topological data analysis for vascular networks is finding meaningful filtrations. So it turns out that, right, we have tumors that are in a mouse model. We have also mathematical models, human tissue. All of these create some point cloud or some network. And then we want to understand, right, how to quantify and how to characterize them. So quantifying the shape of the vessels or the tumor. For example, how much the vascularization, how much the tumor is vascularized. The degree of the abnormality within these blood vessels. So they're very bendy. So often if the ones with high tortuosity are maybe more abnormal, more diseased, or more loops, more anastomosis. And then they also have heterogeneity. So just that it doesn't look like a normal blood vessel in the sense that these all seem to be a bit different. Okay, so trying to find meaningful filtrations. Well, we've looked at quite a few different approaches. So this is work that Bernadette did looking at neuronal networks. So where we simulated data from the Kuramoto model, as well as data that we obtained from Danny Bassett's group of motor learning. So how to, for example, learn how to play Guitar Hero. It's not exactly that, but it's something like this. And from fMRI data, we can get a couple time series, create what's called a functional network. So we have, for these brain regions, there's some, what is called a functional connectivity. So it's weighted, and then we have each of these as a weighted network. And then we can apply persistent homology using the weight rank clique filtration, looking at dimensions 0, 1, and 2. And as you can see, it's really difficult to understand what's happening with these barcodes, but then using persistence landscapes, which I'm not going to define, but the idea is just that we can take this, take the barcode, and we can map it into a nice space to then be able to get some averages and do other types of machine learning. So what we found was that the first day someone is learning a new, for example, some motor skill, learning how to play Guitar Hero, the distance of the average between day 1 and day 2, or day 1 and day 3, is much larger than the distance between day 2 and day 3 on average. But this is one of the filtrations, which is the weight rank clique filtration that we looked at. And Bernadette then applied this to a neuronal network of some data we got from experimentalists in Bari and Roche. And this is looking at schizophrenic, siblings of schizophrenic that are healthy, and controls. And here we got 4 time regimes. So then we, again, computed persistent homology. This is a fMRI, and they do a 2-back test, which means that they have to say what was on a card 2 times ago. And schizophrenic patients often don't perform very well on this. And anyway, so they have the fMRI recorded, and then Brenda analyzes for the 4 different time regimes. And we found that we can split them into controls, patients, and siblings. And so we have that paper on arXiv. So this is all using the weight rank clique filtration. And what was really interesting here is that we found with the fMRI data, it was kind of the shorter bars, so it's those short-lived features that were the discriminating for the neuronal network. Okay, and then other filtrations we've looked at are filtrations in brain artery trees. And Ezra's here, so I'm not gonna, I don't want to say something wrong, but the idea is they have this sweeping filtration from this magnetic resonance angiography. So, and they found the tortuosity, so the bendiness of the brain arteries correlates with the age of the subjects. And so we had tried this filtration with our blood vessel networks. So we considered that. So we've looked at quite a few different filtrations. I'll continue, but so for each of these, we're trying to determine what is the appropriate filtration. I think this is a kind of reoccurring theme, actually, is what is an appropriate filtration for the application. So then some work by Belch et al. This is with Jacek Brodsky. It's looking at COPD, and in particular, he has a sweeping plane filtration on the airways, and this is these are chest scans of these lungs, and they're able to distinguish between different types of patients with this filtration. And then another filtration that's called the radial distance, the radial filtration in neuronal trees here is that they have this, and we ended up using this as a second filtration that we looked at for our problem. And again, you're looking at filtering by this radial distance from to the root of the neuronal tree. So that gives them some difference between the structure of different groups of neurons. Okay, so what we've tried for vascular networks. So all the experimentalists said branching points. This is where you have blood vessels, and it's where they split. And so each of those points, because these are very large images, they could record each of these branching points for us. And so this was the initial data that Bernadette received, was where each of these branching points. And then we just did a Vittori's rips on this. And so here you can see the dimension 0 and the one-dimensional loops. And that's just kind of a small example, because the real example in the micro CT, what we found was that the 0-dimensional weren't that informative, but one-dimensional loops and these voids and the Vittori's rips gives you an idea of how kind of vascularized, how infiltrated the vasculature is in the tumor. So that might give an idea of kind of how diseased. And so we found that the branching points, if you're looking at where the tumor is inside here, often are on the outside, and that the experimentalists we're working with are very happy to see that, because it appears that that's indeed what they see as well. So all of this is static that I've showed you. The other type of filtration that we've been looking at is just defining a center of a tumor and then increasing radially the distance stepwise. So we include all the points within the radius, and so you can kind of see this would be maybe the tumor, and as you're increasing the radius. And so we're able to capture these topological features with respect to the very center of the tumor. And often in the center of the tumor is a lot of the cells are dead. It's necrotic tissue. So so what we found was that this filtration, the dimension 0, gives bendiness of how tortuous, the tortuosity of the blood vessels, but then the dimension 1 gives a flavor of kind of where these loops are here. You can see, and these loops appear approximately halfway between the tumor center and the outside of the tumor. So they're definitely not in the center, but they're not really far away, which again matches what many of the mathematical models have. But what we would like to do is actually have a way of, and that's kind of an active investigation, is being able to quantify the models and quantify, take that kind of our topological summary from model simulated data and actual experimental images that are, these are really beautiful images, and being able to compare them. So now I went very quickly, good. Okay, so is there any questions on that before I continue?
I had a question, Heather. So the one of the last filtrations you described where you're taking these balls out from the center, you just have maybe a fixed complex or a fixed scale for your RIPS complex, and you're including the vertices that are in that growing ball, and then you have a fixed scale.
So what we have is, right, so what you have is that we have this radial distance, and we only include the points once it gets to that distance. And so that's how we're creating this complex.
Great, thanks.
So, I mean, it sounds really obvious now that I've gone through, but actually Bernadette looked at this for a really long time, and when she presented, said, well, what about if I do this? I was like, you know, I mean, it's research, but it's just getting kind of that insight, and that's what it is figuring out for that application what is the appropriate filtration. And this seems to be the experimentalists are really happy. So we've talked with them recently, and they were really happy. So this is all very new work, and I mean, it's motivating kind of the next part of the talk as well.
Great, thanks.
Great. So right, so this part of the talk is available on arXiv, and so the details are there. But the persistent homology pipeline that I've just talked about is that you have some data, right? And then you go to a filtered space, and then you have a persistence module. And so from your finite metric space to then to your filtered simplicial complex. And then here we have, right, we have our filtered simplicial complex to our persistence module, and we get this tuple where we have composition of these maps. And this again is all review. This is the Pth persistent homology of the complex. And so to motivate why we might want to look at multiple parameters, which I believe a couple other people in this kind of series recently have spoken about multi-parameter persistent homology, but let's suppose we have some images like ones I've shown where each of these, we have some grid, and each pixel has some RGB value, for example. So this would be, instead of a bifiltration, it would actually be a trifiltration. And for even small values of, this would be the red and this would be the green, where once we see large values of red, we would see the kind of outside square appearing. But for smaller values, you would see this inside. And eventually it would get, it would fill in, so we would have nothing that lives forever in, for example, dimension 1, but we would see something that lives forever here and here. Okay, that's one example. Another one is these complex biological data sets. So these are the blood vessels that we were just talking about. And this is from the radiation oncology wall, whereas we have other data from Roche. And then another thing where we have this is the different diameter of the thickness of the vessels. But as I said, we have blood vessels that are changing in time as well. And so we have new vasculature appearing. So it would be really nice to have a filtration where we can see actually what's the growth of the vessels in time. So it's taken a long time just to even get a single parameter filtration But I think that there's many different parameters that are of interest to look at in these systems. And finally, one can think about some type of outliers in the data. So instead of just some small measurement noise where you have some real circle here, you might have a couple outliers. For one reason or another, maybe the measurement was taken incorrectly rather than just some measurement noise. Okay, so now if we're looking at multi-parameter persistent homology, the pipeline is we might have data that depends and form an R parameter persistence module. So if we have, we would, going from data to multi-filtered spaces, we would have that partial order on NR. This is the set of R tuples of the natural numbers. And so what we find is that for any pair of elements, U and V, we would define that U is less than V, where UI is less than VI for all these I equals 1 to R. And then we would get some map, whether it's X to R, which would be an R filtered simplicial complex, or these digital images. So then and the next step is going from the multi-filtered space to this multi-parameter persistence module. And so here we have this R parameter persistence module, and this is a tuple with your persistence module, and this is a K vector space. And then for every, again, U less than V, we have these K linear maps where a composition holds. So in other words, you have this R parameter persistence module is a functor from the poset category of NR to the category with objects of K vector spaces and morphisms, the K linear maps. But now on, I think we're going to be a little more hands-on and just talk about some of the algebra here. So we want to know what kind of objects these multi-parameter persistence modules are. So if we put a grading on our polynomial ring, so this is if there's some collection of abelian groups, such as an underlying abelian group is written as this direct sum, and so we have this natural grading where we have this R tuple, and then so we have this isomorphism of categories, and this is from 2009, so almost 10 years ago, but between the category of these R parameter persistence modules and R graded modules over this polynomial ring. But then, of course, there's this problem, which is that there's no decomposition that's analogous in the one parameter case. But I hope that, all is not lost. So then what we have is any persistence module, right, is a homology of a filtered space. So we have this multi-filtered space over this polynomial ring, a multivariate polynomial ring, and any finitely generated NR graded module, there exists a finite multi-filtered simplicial complex and a positive natural number I, such that M is the homology in degree I of this complex. So studying this homology of this R filtered space is studying this graded modules over polynomial rings. Okay, so I think this is just a really nice example from Carlson and Marodian's paper. It's a shame that I can't show you things because I've actually have written out stuff here, but that's fine. I'll just kind of, you can see the mouse, right? Okay, great. So if we think of this as like x1 and this is x2, what we can write is, you can think about these differentials. They're basically, this would be like AB. So if we do it in lexicographic ordering, we have AB and AF and BC and BD, CD, CE, DE, EF. And I would hold all this up to you, but that's okay. So from this bifiltered, this bifiltration, for example, if we want to see what is happening with AB, we would look at, this is AB here. So you see that A is born in, A is born here, and that you're going up one, so you want to have B minus A, and so this would be just going up, so it's x2, whereas B here, before it enters, then it's x1 and then x2 squared. Okay, but let's say we want to see where the homology, we have where it lives. So we have a free summand. We have this being born here in 2, and living forever, right? But then what other things? What else do we see here for first homology dimension? For each one, we see, for example, a triangle is born here, but then dies here. So it's born in 1, but then it's killed in 3, right? And then we see the same thing. There's a triangle born here, but killed in 1 step. So so what we can see is that this feature, we have this generator that lives forever, and then this one is killed. This is killed. Kind of, you have to see it dies, but in the x1 direction, 2 steps, and then it dies. This one dies again in the x1 direction. So I haven't gone through all of it, but hopefully that gives you a flavor. And then there's a lot of algebra background, which I am not going to be able to cover, but I hope that this is motivated, that we want some invariants for applications that are computable, stable, and interpretable, that would show, for example, when things are living, and also, for example, if they live in one, not just things that live forever, but maybe live within one direction. And so this is from their paper. Carlson and Moretti said, "Our study of multi-graded objects shows that no complete discrete invariant exists for multidimensional persistence, and we discriminate invariant that captures persistent information that has homology classes with large persistence." And so this is kind of a summary. I'm noticing I don't have a lot of time left, but because it's 50 minutes, so I have 5 minutes left. But this is a summary that when you're looking at multi-parameter persistent homology, the commutative algebra kind of computation that one would want is, for example, this is very coarse, but computing the rank of the module, which we show in our paper, so you can look at the Hilbert series or the CP rank, and this is really how many things are living in what direction. So, for example, only living in X or Y or in both directions. So you're looking at the rank of the quotient of a module. So this is kind of the summary, but now let me go through some of the details. So I've assumed, I have 2 pages of algebra definitions to actually get to this, so I'm happy to discuss offline as well. Right, but so let me just kind of talk, the major thing, there's the first thing is kind of what is the support shape of our module. So if we assume it's a module over a commutative ring, and then we let U be these non-empty subsets of M, we can define this annihilator as a set of elements of a ring that kill off all elements in this subset. And so then a prime ideal is associated to M if this prime ideal is the annihilator of an element of M. So yeah. So then we can denote the associated primes. This is the poset of all the primes associated to M. And so there's other things I could say, but just in the interest of time, what I think we have as a lemma in our paper is that for a finitely generated NR-graded module M, any associated prime P of M has this form that that this is just a subset of the variables. And so we then we define, it's just a definition that for such a prime P, we can define the complement support. And these are so this is the complement support that we have U1 and 2R in NR, and these are 0 for all of these and then we have also that the support shape of M is then the union of these associated primes of the complement support. So I think it's very quick, but so it's very quick, but then we get the point is that we can say that MPH modules are supported on some union of coordinate hyperplanes, and so we use embedded, so these are non-minimal associated primes to get a stratification of the support shape of the module. And so if we're given some nested sequence of these associated primes, it induces a nested sequence of complement supports. And then, I jumped. The support shape of the module is completely determined by these associated primes. So I think just it's nice to have an example because I'm supposed to be done right now. So if we have two different modules here, M and N, what we find is since these are free, the associated primes at 0, so you get the whole. This is the So you get all of these points where the module doesn't vanish. And this is the same for N, right? Because we also have this, but the shape is actually here. And then what I want to continue is specifically because we have these parts where things only live in some of the you don't have the whole thing. We can see where we don't have that associated prime, that we don't have the 0 ideal. We have either things that are shifted to the origin. So we have, for example, this is the complement support where you just have the origin, or you only have this coordinate plane, or the whole space. And then if you're looking at for X2, again, we see just this kind of the stratification, these chains. So, okay, so then what we would see in the colored images is some feature that lives in H1, for example, that lives across here, and so for X and Y, for example. Sorry, I don't know. Just hang on. Okay, so in the interest of time, so we can generalize the definition of birth and death, but I won't go into that. I hope I gave you just a bit of a flavor, but the idea is that you're looking at the radical of this annihilator, and then we can kind of split this up. The support, the elements of the support dimension for 0 are transient, for 1 are partially persistent or fully persistent, and all of this, there's some information that gets forgotten. For example, we don't see that this is the translation. Let me see, is that shown? Oh, so you would only see that you wouldn't get that this, for example, there's a thickness or that you have this multiplicity. But then if we're looking at the rank of the zeroth local cohomology, then we can capture some of this. That, for example, this is rank 1, and then this is rank 2, and this is rank 2. So we get some of the information. So thanks for listening. I'm going to very quickly conclude. So what we showed is that the stratification of the support shape gives this summary of elements that are living along certain coordinate subspaces, and the stability is something that that Nina is currently investigating. And then there's a lot of computation. So all our Macaulay 2 code, which I had put in as like screenshots, but then as I was practicing, this was going way over time, which is why I kept taking out some algebra as well. But all of that's available in our preprint. I think Nina's writing a package. Oliver Vipon and Parker Edwards. So Oliver's generalized multi-parameter persistence landscapes, and I believe that they're working on a package, and Oliver's persistence, multi-parameter persistence landscapes have given us some really nice insight into another problem related to cancer. And so then there's a lot of other work, which is just really, I'm not including, I mean, I could go a whole slide, but I just wanted to put in a couple different invariants and approaches that people are using for studying multi-parameter persistence modules. Okay, and finally, I need to make a plug. So the SIAM Journal of Applied Algebra and Geometry are very interested in applied topology for pure type of papers. So if you have a manuscript where you're using some ideas from pure math, for example, normally algebra and topology is not that prevalent in biomath journals, but they're very open to it in letters. And then there's a new journal, Foundations of Data Science, which is starting in January. And again, they're very interested in applied topology. So I thought I would make a plug for that. And also, we have two postdocs that we're looking to hire, and it closes today. So if you're interested, please apply. And finally, I really have to thank Nina and Bernadette. I have kind of shamelessly used a lot of their slides here, and they've done a lot of great work. And finally, thank you.
Great, thank you, Heather. Does anyone have any questions? If you do, then unmute yourself. Okay, so I have one question. Because you already mentioned, I mean, you have sort of a version of software. Have you actually tried any, like, anything on a real data set, or like, is it so far it was mostly like toy examples of?
So it turns out, right? So I know that that Mike and Matthew have Rivet, where they can have this bifiltered simplicial complex, but it turns out trying to enter that into Macaulay 2 is really, really, really hard. There's no easy way to do that, as far as I'm aware. If there is, I mean, I've spoken with a couple of people that write Macaulay 2 packages as well, and it's a really hard problem. So that's like kind of what we've developed is kind of, we've proposed a lot of different information about these MPH modules, right? Different invariants that one can compute depending on what information they're interested in. So we don't propose just one invariant in our paper because, I mean, first we need to work with real data sets and getting them into a bifiltered simplicial complex or multi-filtered into Macaulay 2 is actually really hard. So what we've shown are really toy, where kind of by hand we have to enter everything in, and that's for these multi-degrees and everything. It's not very nice, but I'm very open. If somebody has something already, or that would be really great.
Okay, great.
So the answer is no, not yet.
Yeah. Okay, great. So if there are no questions, then I would
let me ask another question.
Okay, very good.
If that's all right. Heather, is it fair to say that, or is it correct to say that in the one-dimensional case, do the stratifications of the support shapes, do they recover the one-dimensional intervals?
Well, you have with the what you have with the one-dimensional, right? You can actually write out, for example, the Hilbert series, and you would see that you have two features that live forever in that example, which I didn't write, that I couldn't show you, but I specifically done this for one parameter because I thought it would be nice to give an example. So so you can see the features that live forever in that but you have this complete this decomposition. So it's I mean, it's just all the information in that module.
Great, thanks.
Okay, so in case there are no other questions, I would like to ask everyone to unmute themselves so that we can clap for Heather.
Oh, thanks, everybody. Thanks.
Okay, thanks again, everyone, for participating. And so the video should appear online shortly. And because this is the last seminar of the semester, I would like to wish all of you nice holidays. And I guess we'll be back in January. Yeah. Thank you.